In May 2000, just weeks before the announcement of the first draft of the Human Genome, a notebook began circulating among the scientists sitting at the bar in Cold Spring Harbor Laboratory in New York State. Excitement was building about the next phase of the Human Genome Project, in which the DNA sequence would be split into its functional parts, genes. The notebook contained a sweepstake, the guesses of the best-informed group of people on the planet, concerning one intriguing question: how many genes does it take to build a human? Senior research scientist Lee Rowan, who was leading a group working on decoding chromosomes 14 and 15, sipped her beer as she pondered the question. Genes make proteins, the building blocks of life, and the sheer complexity of humans made it seem probable that the number would be high, higher than the mouse, surely, which was known to have 23,000 genes, probably also higher than the wheat plant with 26,000 genes, and no doubt far higher than the worm, a favorite laboratory species of developmental biologists, with its 20,500 genes, despite guesses averaging over 55,000 genes and topping 150,000. Rowan's understanding of the field meant she was inclined to go low. She placed a bet of 41,440 that year, and followed it up a year later with a second bet of just 25,947 genes. In 2003, with the true gene number only just emerging from the nearly finished sequence, Rowan was awarded the prize. Her entry was the lowest of all 165 bets, and the latest gene count had just dropped even lower than any scientist had ever predicted. With just shy of 21,000 genes, the human genome is hardly bigger than that of the worm C. elegans. It is half the size of the rice plant, and even the humble water flea outstrips it with 31,000 genes. None of these species can talk, create, or think intelligent thoughts. You might think, as the scientists entering the genosweep pool did, that humans would have a great many more genes than grasses and worms and fleas. After all, genes make proteins, and proteins make bodies. Surely a body as complex and sophisticated as a human's would need more proteins, and therefore more genes, than a worm's. But these 21,000 genes are not the only genes that run your body. We do not live alone. Each of us is a superorganism, a collective of species, living side by side and cooperatively running the body that sustains us all. Our own cells, though far larger in volume and weight, are outnumbered 10 to 1 by the cells of the microbes that live in and on us. These 100 trillion microbes, known as the microbiota, are mostly bacteria, microscopic beings made of just a single cell each. Alongside the bacteria are other microbes, viruses, fungi, and archaea. Viruses are so small and simple that they challenge our ideas of what even constitutes life. They depend entirely on the cells of other creatures to replicate themselves. The fungi that live on us are often yeasts, more complex than bacteria but still small single-celled organisms. The archaea are a group that appear to be similar to bacteria, but they are as different evolutionarily as bacteria are from plants or animals. Together, the microbes living on the human body contain 4 million genes. This is the microbiome, the collective genomes of the microbiota. These genes collaborate in running our bodies alongside our 21,000 human genes. By that count, you are just 0.5% human. We now know that the human genome generates its complexity not only in the number of genes it contains, but also through the many combinations of proteins these genes are able to make. We and other animals are able to extract more functions from our genomes than they appear to encode at first glance. But the genes of our microbes add even more complexity to the mix, providing services to the human body that are more quickly evolved and more easily provided by these simple organisms. Until recently, studying these microbes relied on being able to culture them on petri dishes filled with broths of blood, bone marrow, or sugars, suspended in jelly. It's a difficult task. Most of the species living in the human gut die on exposure to oxygen. They simply haven't evolved to tolerate it. What's more, growing microbes on these plates means guessing what nutrients, temperature, and gases they might need to survive, and failing to figure this out means failing. To learn more about a species, culturing microbes is the equivalent of checking who's turned up for class by running down a register. If you don't call someone's name, you won't know if they are there. Today's technology, the DNA sequencing made so fast and cheap by the efforts of those working on the Human Genome Project, is more like requesting ID at the door. Even those that you weren't expecting can be accounted for. As the Human Genome Project came to a close, expectations were high. It was seen as the key to our humanity, God's greatest work, and a sacred library holding the secrets of disease. When the first draft was completed in June 2000, under budget at $2.7 billion and several years early, the U.S. President Bill Clinton declared, "Today we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases." But in the years that followed, science journalists the world over began expressing their disappointment in the contribution that knowledge of our complete DNA sequence had made to medicine. Although decoding our own instruction book is an irrefutable achievement that has made a difference to treatments for several important illnesses, it has not revealed as much as we expected about the causes of many common diseases. Searching for genetic differences in common to people with a particular disease did not throw up straightforward links for as many conditions as had been expected. Often, conditions were weakly linked to tens or hundreds of gene variants, but rarely was it the case that possessing a given gene variant would lead directly to a given disease. What we failed to appreciate at the turn of the century was that those 21,000 genes of ours are not the full story. The DNA sequencing technology invented during the Human Genome Project enabled another major genome sequencing program, but one that received far less media attention, the Human Microbiome Project. Rather than looking at the genome of our own species, the HMP was set up to use the genomes of the microbes that live on the human body, the microbiome, to identify which species are present. No longer would a reliance on petri dishes and an overabundance of oxygen hold back research into our cohabitors. With a budget of $170 million and a 5-year program of DNA sequencing, the HMP was to read thousands of times as much DNA as the HGP, from microbes living in 18 different habitats on the human body. It was to be a far more comprehensive survey of the genes that make a person both human and microbial. At the conclusion of the Human Microbiome Project's first phase of research in 2012, not one world leader made a triumphant statement, and only a handful of newspapers featured the story. But the HMP would go on to reveal more about what it means to be human today than our own genome ever has. Since life began, species have exploited one another, and microbes have proved themselves to be particularly efficient at making a living in the oddest of places. At their microscopic size, the body of another organism, particularly a macro-scale backbone creature like a human, represents not just a single niche, but an entire world of habitats, ecosystems, and opportunities, as variable and dynamic as our spinning planet. The human body has a chemical climate that waxes and wanes with hormonal tides, and complex landscapes that shift with advancing age. For microbes, this is Eden. We have been co-evolving side by side with microbes since long before we were humans, before our ancestors were mammals even. Each animal body, from the tiniest fruit fly to the largest whale, is yet another world for microbes. Despite the negative billing many of them get as disease-causing germs, playing host to a population of these miniature life forms can be extremely rewarding. The Hawaiian bobtail squid, as big-eyed and colorful as any Pixar character, has diminished a major threat to its life by inviting just one species of bioluminescent bacterium to live in a special cavity in its underbelly. Here, in this light organ, the bacteria known as Vibrio fischeri convert food into light so that viewed from below the squid glows. This obscures its silhouette against the moonlit ocean surface, camouflaging it from predators approaching from beneath. The squid owes this protection to its bacterial inhabitants, and they owe the squid for their home. While housing a microbial light source might seem a particularly inventive way to increase one's life chances, squid are far from the only animal species who owe their lives to their body's microbes. Strategies for living are many and varied, and cooperation with microbes has been a driving force of the evolutionary game since living beings with more than one cell first evolved, 1-2 billion years ago. The more cells an organism is made of, the more microbes can live on it. Indeed, large animals such as cattle are well known for their bacterial hospitality. Cows eat grass, yet using their own genes they can extract very little nutrition from this fibrous diet. They would need specialist proteins called enzymes that can break down the tough molecules, making the cell walls of the grass. Evolving the genes that make these enzymes could take millennia, as it relies on random mutations in the DNA code that can only happen with each passing generation of cows. A quicker way to acquire the ability to get at the nutrients locked away in grass is to outsource the task to the specialists, microbes. The four chambers of the cow's stomach house populations of plant fiber-busting microbes, numbering in their trillions, and the cud, a ball of solid plant fiber, travels back and forth between the mechanical grinding of the cow's mouth and chemical breakdown by the enzymes produced by microbes living in the gut. Acquiring the genes to do this is quick and easy for microbes, as their generation times and therefore opportunities for mutations and evolution are often less than a day. If bobtail squid and cows can both benefit from teaming up with microbes, is it possible that we humans do as well? We may not eat grass and have a four-chambered stomach, but we do have our own specializations. Our stomachs are small and simple. They're just to mix the food up, throw in some enzymes for digestion, and add a bit of acid to kill unwelcome bugs, but travel on through the small intestine, where food is broken down by yet more enzymes and absorbed into the blood through the carpet of finger-like projections that give it the surface area of a tennis court, and you reach a cul-de-sac, more of a tennis ball than a tennis court, that marks the beginning of the large intestine. This pouch-like patch at the lower right corner of your torso is called the cecum, and it is the heart of the human body's microbial community. Dangling from the cecum is an organ that has a reputation for being there simply to cause pain and infection, the appendix. Its full title, the vermiform appendix, refers to its worm-like appearance, but it could equally be compared to a maggot or a snake. Appendices vary in length from a diminutive 2 cm to a distinctly stringy 25 cm, and rarely, a person may even have two of them or not one at all. If popular opinion is to be believed, we would be better off without one at all, since for over 100 years they have been said to have no function whatsoever. In fact, the man who finally put the anatomy of animals into an elegant evolutionary framework is apparently responsible for this persistent myth. Charles Darwin in The Descent of Man, a follow-up to On the Origin of Species, included the appendix in a discussion of rudimentary organs. Having compared it with the larger appendices of many other animals, Darwin felt that the appendix was a vestige, steadily withering away as humans changed their diets. With little to indicate otherwise, the vestigial status of the appendix was barely questioned for the next 100 years, and the perception of its uselessness is only enhanced by its tendency to cause a nuisance. So pointless has the medical establishment assumed it to be, that by the 1950s, removing it became one of the most common surgical procedures carried out in the developed world. An appendectomy was even often tacked on as a bonus during other abdominal surgery. At one point, a man stood a 1 in 8 chance of having his appendix removed during his lifetime, and for a woman the odds were 1 in 4. About 5–10% of people get appendicitis at some stage in their life, usually in the decades before they have children. Untreated, nearly half of these people would die. This presents a conundrum. If appendicitis were a naturally occurring disease frequently causing death at a young age, the appendix would be quickly eliminated by natural selection. Those with appendices large enough to become infected would die, most often before reproducing, and would therefore fail to pass on their appendix-forming genes. Over time, fewer and fewer people would have an appendix, and eventually it would be lost. Natural selection would have preferred those without one. Darwin's assumption that it was a relic of our pasts might have carried some weight were it not for the often fatal consequences of possessing one. There are two explanations, therefore, for the persistence of the appendix, and they are not mutually exclusive. The first is that appendicitis is a modern phenomenon brought on by some environmental change. Thus, even a pointless organ could have persisted in the past simply by keeping out of trouble. The other is that the appendix, far from being a malign vestige of our evolutionary past, actually has health benefits that outweigh its dark side, making its presence worthwhile despite the risk of appendicitis. That is, natural selection prefers those of us who possess one. The question is why? The answer lies in its contents. The appendix, which averages about 8 cm in length and a centimeter across, forms a tube protected from the flow of mostly digested food passing its entrance, but rather than being a withered strand of flesh, it is packed full of specialized immune cells and molecules. They are not inert, but rather an integral part of the immune system, protecting, cultivating, and communicating with a collective of microbes. Inside, these microbes form a biofilm, a layer of individuals that support one another and exclude bacteria that might cause harm. The appendix, far from being functionless, appears to be a safe house that the human body has provided for its microbial inhabitants. Like a nest egg stashed away for a rainy day, this microbial stockpile comes in handy at times of strife. After an episode of food poisoning or a gastrointestinal infection, the gut can be repopulated with its normal inhabitants, which have been lurking in the appendix. It might seem like an excessive bodily insurance policy, but it is only in recent decades that gut infections such as dysentery, cholera, and giardiasis have been all but eliminated in the Western world. Public sanitation measures, including sewerage systems and water treatment plants, have prevented such illnesses in developed countries, but globally, 1 in 5 of all childhood deaths are still caused by infectious diarrhea. For those who do not succumb, possession of an appendix likely hastens their recovery. It is only in a context of relatively good health that we have come to believe that the appendix has no function. Indeed, the negative consequences of undergoing an appendectomy have been masked by the modern sanitized lifestyle. As it turns out, appendicitis is a modern phenomenon. In Darwin's day, it was extremely rare, causing very few deaths, so we can perhaps forgive him for thinking the appendix was merely one of evolution's leftovers, neither harming nor helping us. Appendicitis became common in the late 19th century, with cases in one British hospital shooting up from a stable rate of 3 or 4 people per year prior to 1890 to 113 cases per year by 1918, a rise mirrored throughout the industrialized world. Diagnosis had never been a problem. The cramping pain followed by a quick autopsy, if the patient didn't make it, revealed the cause of death even before appendicitis became as common as it is now. Many explanations were put forward to explain it, from increased meat, butter and sugar consumption, to blocked sinuses and rotting teeth. At that time, consensus opinion alighted on a reduction in fiber in our diets as the ultimate cause, but hypotheses still abound, including one that blames the rise on improved water sanitation and the hygienic conditions it brings, the very development that appeared to render the appendix almost impotent. Whatever the ultimate cause, by the Second World War, our collective memory had been purged of the rise in appendicitis cases, leaving us with the impression that it is an expected though unwelcome feature of normal life. In fact, even in the modern, developed world, keeping hold of the appendix, at least until adulthood, can prove to be beneficial, protecting us from recurring gastrointestinal infections, immune dysfunction, blood cancer, some autoimmune diseases, and even heart attacks. Somehow its role as a sanctuary of microbial life brings these benefits. That the appendix is far from pointless tells us something bigger. Our microbes matter to our bodies. It seems they are not just hitching a ride, but providing a service important enough that our guts have evolved an asylum just to keep them safe. The question is, who is there and what exactly do they do for us? Although we have known for several decades that our body's microbes offer us a few perks, like synthesizing some essential vitamins and breaking down tough plant fibers, the degree of interaction between our cells and theirs wasn't realized until relatively recently. In the late 1990s, using the tools of molecular biology, microbiologists took a great leap into discovering more about our strange relationship with our microbiotas. New DNA sequencing technology can tell us which microbes are present and allows us to place them within the tree of life, with each step down this hierarchy from domain to kingdom, then phylum through class, order and family, and on to genus, species, and strain. Individuals are more and more closely related to one another. Working from the bottom up, we humans, genus Homo, and species sapiens, are great apes, family Hominidae, which sit alongside monkeys and others within the primates, order primates. All of us primates belong with our fellow furry milk drinkers as a member of the mammals class Mammalia, who then fit within a group containing animals with a spinal cord, phylum Chordata, and finally, amongst all animals, spinal cord or otherwise. Think of our squid, for example, in kingdom Animalia and domain Eukarya. Bacteria and other microbes, except the category-defying viruses, find their place on the other great branches of the tree of life, belonging not to kingdom Animalia, but to their own unique kingdoms in separate domains. Sequencing allows different species to be identified and placed within the hierarchy of the tree of life. One particularly useful segment of DNA, the 16S rRNA gene, acts as a kind of barcode for bacteria, providing a quick ID without the need to sequence an entire bacterial genome. The more similar the codes of the 16S rRNA genes, the more closely related the species, and the more twigs and branches of the tree of life they share. DNA sequencing, though, is not the only tool at our disposal when it comes to answering questions about our microbes, especially regarding what they do. For these mysteries, we often turn to mice, in particular germ-free mice. The first generations of these laboratory staples were born by cesarean section and kept in isolation chambers, preventing them from ever becoming colonized with microbes, either beneficial or harmful ones. From then on, most germ-free mice are simply born in isolation to germ-free mothers, sustaining a sterile line of rodents untouched by microbes. Even their food and bedding is irradiated and packed in sterile containers to prevent any contamination of the mice. Transferring mice between their bubble-like cages is quite an operation, involving vacuums and antimicrobial chemicals. By comparing germ-free mice with conventional mice, which have their full complement of microbes, researchers are able to test the exact effects of having a microbiota. They can even colonize germ-free mice with a single species of bacterium or a small set of species to see precisely how each strain contributes to the biology of a mouse. From studying these notobiotic known life mice, we get an inkling of what microbes do in us as well. Of course, they are not the same as humans, and sometimes results of experiments in mice are wildly different from results in humans, but they are a fantastically useful research tool and very often provide crucial leads. Without rodent models, medical science would progress at a millionth of the speed. It was by using germ-free mice that the commander-in-chief of microbiome science, Professor Jeffrey Gordon from the University of Washington in St. Louis, Missouri, discovered a remarkable indication of just how fundamental the microbiota are to the running of a healthy body. He compared the guts of germ-free mice with those of conventional mice, and he learnt that under the direction of bacteria, the mouse cells lining the gut wall were releasing a molecule that fed the microbes, encouraging them to take up residence. The presence of a microbiota not only changes the chemistry of the gut, but also its morphology. The finger-like projections grow longer at the insistence of microbes, making the surface area large enough to capture the energy it needs from food. It's been estimated that rats would need about 30% more to eat if it weren't for their microbes. It is not only microbes that are benefiting from sharing our bodies, but us too. Our relationship with them is not just one of tolerance, but encouragement. This realization, combined with the technical power of DNA sequencing and germ-free mouse studies, began a revolution in science. The Human Microbiome Project, run by the United States National Institutes for Health, alongside many other studies in laboratories around the world, has revealed that we utterly depend on our microbes for health and happiness. The human body, both inside and out, forms a landscape of habitats as diverse as any on Earth, just as our planet's ecosystems are populated by different species of plants and animals, so the habitats of the human body host different communities of microbes. We are like all animals in an elaborate tube. Food goes in at one end of the tube and comes out the other. We see our skin as covering our outside surface, but the inner surface of our tube is also outside, exposed to the environment in a similar way. whilst the layers of our skin protect us from the elements invading microbes and harmful substances. The cells of the digestive tract that runs right through us must also keep us safe. Our true inside is not this digestive tract, but the tissues and organs, muscles and bones that are packed away between the inside and the outside of our tubular selves. The surface of a human being, then, is not just their skin, but the twists and turns, furrows, and folds of their inner tube as well. Even the lungs, the vagina, and the urinary tract count as being on the outside, as part of the surface when you view the body in this way. No matter if it's inner or outer, all of this surface is potential real estate for microbes. Sites vary in their value, with dense city-like communities building up in resource-rich prime locations like the intestines, while sparser collections of species occupy more rural or hostile locations such as the lungs and the stomach. The Human Microbiome Project set out to characterize these communities by sampling microbes from 18 sites across the inner and outer surfaces of the human body, in each of hundreds of volunteers. Over the first 5 years of the HMP, molecular microbiologists oversaw biotechnological echo of the golden age of species discovery, one that saw formaldehyde-infused collecting cabinets stuffed with the haul of birds and mammals discovered and named by explorer biologists in the 18th and 19th centuries. The human body is, as it turns out, a treasure trove of strains and species new to science, many of them present on only one or two of the volunteers participating in the study, far from each person harboring a replicate set of microbes. Very few strains of bacteria are common to everyone. Each of us contains communities of microbes as unique as our fingerprints. Though the finer details of our inhabitants are specific to each of us, we all play host to similar microbes at the highest hierarchical levels. The bacteria living in your gut, for example, are more similar to the bacteria in the gut of the person sitting next to you than they are to the bacteria on your knuckles. What's more, despite our distinctive communities, the functions they perform are usually indistinguishable. What bacterium A does for you, bacterium B might do for your best friend. From the arid, cool plains of the skin on the forearms to the warm, humid forests of the groin and the acidic, low-oxygen environment of the stomach, each part of the body offers a home to those microbes that can evolve to exploit it. Even within a habitat, distinct niches host different collections of species. The skin, all 2 square meters of it, contains as many ecosystems as the landscapes of the Americas, but in miniature. The occupants of the sebum-rich skin of the face and back are as different from those of the dry, exposed elbows as the tropical forests of Panama are from the rocks of the Grand Canyon, where the face and back are dominated by species belonging to the genus Propionibacterium, which feed off the fats released by the densely packed pores. In these areas, the elbows and forearms host a far more diverse community. Moist areas, including the navel, the underarms, and the groin, are home to Corynebacterium and Staphylococcus species, which love the high humidity and feed off the nitrogen in the sweat. This microbial second skin provides a double layer of protection to the body's true interior, reinforcing the sanctity of the barrier formed by the skin. Cells, invading bacteria with malicious intentions, struggle to get a foothold in these closely guarded bodily border towns and face an onslaught of chemical weapons when they try. Perhaps even more vulnerable to invasion are the soft tissues of the mouth, which must resist colonization by a flood of intruders smuggled on food and floating in the air. From the mouths of their volunteers, researchers working on the Human Microbiome Project took not just one sample, but 9, each from a slightly different location. These 9 sites turned out to have discernibly different communities within mere centimeters of one another, made up of around 800 species of bacteria dominated by Streptococcus species and a handful of other groups. Streptococcus gets bad press on account of the many species that cause diseases from strep throat to the flesh-eating infection necrotizing fasciitis. But many other species in this genus behave themselves impeccably, crowding out nasty challengers in this vulnerable entrance to the body. Of course, these tiny distances between sampling locations within the mouth may seem insignificant to us, but to microbes they are like vast plains and mountain ranges with climates as different as northern Scotland and the south of France. Imagine then the climatic leap from the mouth to the nostrils, the viscous pool of saliva on a rugged bedrock replaced by a hairy forest of mucus and dust. The nostrils, as you might expect from their gatekeeper status at the entrance to the lungs, harbor a great range of bacterial groups, numbering around 900 species, including large colonies of Propionibacterium, Corynebacterium, Staphylococcus, and Moraxella. Heading down the throat towards the stomach sees the enormous diversity of species found in the mouth drop dramatically. The highly acidic stomach kills many of the microbes that enter with food, and just one species is known for certain to reside there permanently in some people, Helicobacter pylori, whose presence may be both a blessing and a curse. From this point on, the journey through the digestive tract reveals an ever greater density and diversity of microbes. The stomach opens into the small intestine, where food is rapidly digested by our very own enzymes and absorbed into the bloodstream. There are still microbes here, though, around 10,000 individuals in every milliliter of gut contents at the start of this 7 m long tube, rising to an incredible 10 million per milliliter at the end, where the small intestine meets the large intestine, starting point. Just outside the safe house created by the appendix is a teeming metropolis of microbes, in the heart of the microbial landscape of the human body, the tennis ball-like cecum to which the appendix is attached. This is the epicenter of microbial life, where trillions of individual microbes of at least 4,000 species make the most of the partially digested food that has passed through round 1 of the nutrient extraction process in the small intestine. The tough bits, plant fibers, are left over for the microbes to tackle in round 2. The colon, which forms most of the length of the large intestine, running up the right-hand side of your torso, across your body under your rib cage and back down the left-hand side, provides homes for microbes, numbering 1 trillion individuals per milliliter by now, in the folds and pits of its walls. Here, they pick up the scraps of our food and convert them into energy, leaving their waste products to be absorbed into the cells of the colon's walls. Without the gut's microbes, these colonic cells would wither and die, whilst most of the body's cells are fed by sugar transported in the blood. The colonic cell's main energy source is the waste products of the microbiota. The colon's moist, warm, swamp-like environment, in parts completely devoid of oxygen, provides not only a source of incoming food for its inhabitants, but a nutrient-rich mucus layer, which can sustain the microbes in times of famine. Because HMP researchers would have to cut open their volunteers to sample the different habitats of the gut, a far more practical way of collecting information about the gut's inhabitants was to sequence the DNA of microbes found in the stool. On its passage through the gut, the food we eat is mostly digested and absorbed both by us and our microbes, leaving only a small amount to come out the other end. Stool, far from being the remains of our food, is mostly bacteria, some dead, some alive, around 75% of the wet weight. Of feces is bacteria. Plant fiber makes up about 17%. At any one time, your gut contains about 15 kg of bacteria. That's about the same weight as the liver, and the lifespans of individuals are a matter of just days or weeks. The 4,000 species of bacteria found in the stool tell us more about the human body than all the other sites put together. These bacteria become a signature of our health and dietary status, not only as a species, but as a society and personally. By far the most common group of bacteria in the stool are the bacteroids, but because our gut bacteria eat what we eat, bacterial communities in the gut vary from person to person. The gut microbes aren't just scavengers, though, taking advantage of our leftovers. We have exploited them too, especially when it comes to outsourcing functions that would take us time to evolve for ourselves. After all, why bother having a gene for a protein that makes vitamin B12, which is essential for our brain function, when Klebsiella will do it for you? And who needs genes to shape the intestine's walls when bacteroids have them? It's much cheaper and easier than evolving them afresh. But as we will discover, the role of the microbes living in the gut goes far beyond synthesizing a few vitamins. The Human Microbiome Project began by looking only at the microbiotas of healthy people. With this benchmark set down, the HMP went on to ask how they differ in poor health, whether our modern illnesses could be a consequence of those differences, and if so, what was causing the damage. Could skin conditions like acne, psoriasis, and dermatitis signal disruption to the skin's normal balance of microbes? Might inflammatory bowel disease, cancers of the digestive tract, and even obesity be due to shifts in the communities of microbes living in the gut? And most extraordinarily, could conditions that were apparently far removed from microbial epicenters, such as allergies, autoimmune diseases, and even mental health conditions, be brought on by a damaged microbiota? Lee Rowan's educated guess in the sweepstake at Cold Spring Harbor hinted at a much deeper discovery. We are not alone, and our microbial passengers have played a far greater role in our humanity than we ever expected. As Professor Jeffrey Gordon puts it, this perception of the microbial side of ourselves is giving us a new view of our individuality, a new sense of our connection to the microbial world, a sense of the legacy of our personal interaction with our family and environment early in life. It's causing us to pause and consider that there might be another dimension to our human evolution. We have come to depend on our microbes, and without them, we would be a mere fraction of our true selves. So what does it mean to be just 10% human?